So, good morning. My name is Jhon Esterilla, and I am medical student at Pontificia Universidad Javeriana in Cali, Colombia. Today, I will be presenting my research, discriminative analysis of the chromosome 13 on DNA repeat composition and functional disease-related genes. So, let's start with the background. The study of human chromosomes is essential to understand the origin of genetic diseases. In this case, chromosome 13 plays a key role in various biological processes and disease mechanisms. However, its genomic structure and the functional significance of its DNA repeat elements are still unexplored. This study aims to characterize chromosome 13 genomic context, focusing on associations with DNA repeat sequences, functional enrichment, and disease-related variants, to better understand potential impacts on gene regulation and chromatin dynamics. Now, materials and methods. To determine In this study, to determine DNA composition of chromosome 13 and analyze how these repeated elements are related to the development of specific pathologies, we employed a bioinformatics approach divided into four stages. Initially, we performed genomic information retrieval of chromosome 13, then the functional enrichment analysis. Subsequently, we defined protein interactions and related processes, and finally, we identified associated diseases. And here we can see some of the platforms we used for this research, like Genome Browser, Enrich, String, NCBI, ShinyGo, and other ones. Mm, Let's talk about the results and discussion. So, during the bibliography research, we found different types of diseases associated with this chromosome, including diseases of central nervous system, digestive system, immune system, among others. Overall, most of the articles related to chromosome 13 associated diseases focus on Patau syndrome and chromosomal aberrations. Now, in protein interaction definition and related processes, we have the figure one that is the chromosome 13 interactome. Basically, this figure shows the functional network analysis of genes on chromosome 13. So we have letter A for complete interactome without clustering and letter B for interactome with clustering. In this case, we identified three clusters, uh, one in blue, the other in red, and the last one in green. These clusters are based on physical proximity and gene-to-gene interactions, which tend to form networks that could contribute to certain biological processes. Despite this clustering, our analysis revealed few connections to major cellular pathways, suggesting that chromosome 13 genes may play a more limited role in primary cellular functions. Now, in functional enrichment, we have table 1, that is the chromosome 13 related diseases based on functional enrichment. Basically, we compare diseases associations for chromosome 13 using two platforms, TopGene and Enrich. TopGene identified statistically significant associations for some phenotypes or for some diseases, while Enrich didn't find significant associations. This discrepancy highlights a lack of consensus of chromosome 13 diseases associations, emphasizing the need for further research. Now, in genomic characteristics of chromosome 13 genes, we explore the number and types of variants on chromosome 13 based on descriptions found in Variation Viewer database. In Figure 2, we can see the analysis by quartiles for clinic variations related to diseases, clean bar yes for Q2 for second quartile and third quartile, and the minor allele frequency, also known as MIF, for second quartile and third quartile, while statistical analysis for most disease-associated variants reveal no significant enrichment compared to the background of genome. Here we can see. And the low MIF values might indicate a scarcity of allelic variants contributing to rare diseases. So basically, the limited statistical association between genetic variants of chromosome 13 and their manifestations in pathological processes, the findings indicate limited evidence for high pathogenicity. Conversely, there is a stronger evidence for low pathogenicity, suggesting influence of indirect genomic factors in disease onset. In conclusion, this work provides an in-depth genomic characterization of chromosome 13, highlighting regions of functional and clinical significance. These findings enhance the understanding of chromosome 13 architecture and suggest influence in regulatory networks and disease mechanisms, offering new insights for therapeutic exploration. Here we have the reference. Thank you very much. That's everything.